I would like to welcome Dr.Teri Manolio to give us the keynote lecture. I wish that by learning from Dr. Manolio's experience today, Thailand will be given a boost to move forward on implementing genomic medicine into our healthcare system to benefit our Thai people and transforming the healthcare industry. Thank you so much.
And now today I have an opportunity to learn about many other things that you're doing in genetics and genomics. But I thought I would take some time to tell you a little bit about what we're doing in the U.S. that might be of interest to you and things that you might like to do in addition. So I should show you this is sunrise in Washington, D.C. It'll happen in about 5 hours from now, so forgive me if I'm not totally coherent, but I will do my best. I thought I would talk to you a little bit about how we at the National Human Genome Research Institute, which is one of the 27 institutes and centers of the National Institutes of Health, how we define genomic medicine. And then some activities of a working group that we have established to advise us on how best to move forward in this area. And then I'll describe some of our major programs and some intriguing findings from them. So we began moving down this path about nine years ago now, when we developed a strategic plan. This is a sort of a roadmap that each of the institutes develops, usually every 7 or 8 years or so. This is going to be exciting. Okay. Every 7 or 8 years ago to try to understand what its research directions should be. This was published in the journal Nature. And one of the things we wanted to determine was how can we best use genomics to identify risk, prevent disease, improve diagnostics, improve treatments, and increase access to care. So we see a spectrum of genomics research. A large part of the efforts that we do have to do with discoveries, so identifying relationships between genetic variation and various phenotypic characteristics, identifying people at risk for disease, finding all the variants associated with the disease, that sort of thing. And there's clinical validation, which is sort of assessing the outcomes after using genetics to direct therapy. And you'll find that I use the terms genetics and genomics pretty much interchangeably. Francis Collins, who's the director of the NIH and previously was director of our institute, is fond of saying that the only difference between genetics and genomics is 2 letters. We tend to think of genetics as being more single gene diseases or conditions, and genomics as being things that are multifactorial with multiple genes and the environment affecting them. And then we also see clinical implementation as a major effort, developing the processes for performing genetic and genomic testing and using the results in clinical care. At NHGRI, we see genomic medicine as being sort of these latter 2 efforts, and that's how we use the term. We have an advisory group, a working group that comes together actually every month by conference call, and then once a year for an all-day meeting in Bethesda, and then periodic meetings throughout the year. You can see the members here. They do a tremendous job in giving us outstanding advice, and then several of us in the effort as well. And one of the things we did early on with this group was to come up with a definition of genomic medicine, which we define as an emerging medical discipline that involves using genomic information about an individual as part of their clinical care. So not just genetic information that we're aware of from studies of large numbers of people, but actually measuring an individual's genome and then using that information. We charged this working group with assisting and advising us on research needed to evaluate and implement genomics in clinical care, to review current progress, identify research gaps and approaches for filling them, identify and publicize key advances in genomic medicine, develop and plan genomic medicine meetings that are on relevant themes periodically, facilitate collaborations and coordination, and consider how we can sustain these efforts. One of the things that we do on each of our conference calls is to review the current literature and identify papers that we see as being evidence that genomic medicine is effective and should be used in clinical care. And we publish these on our website in this heading here, Notable Accomplishments in Genomic Medicine. And if you click on that, you'll then see a series of topics in clinical implementation. For example, some months ago we had this paper from our Ignite network that was felt to be a major advance. And if you click on that, you can open it up, find the paper. And here's another one more recently, the CARA paper on polygenic scores. So this is a resource. If people ask you, what is it that genomic medicine is most actively doing, this might be a place to look. And this is available and free to everyone, so I encourage you to use it if you'd like. Another thing, as I mentioned, that this group does is to plan periodic meetings. These are usually workshops of 100, 120 people or so that we invite to Bethesda and ask for their views over a 2-day period on a given topic. So one topic initially was sort of, what genomic medicine research is going on out in the U.S. That was our very first meeting. The second one focused on collaborations. We had a third one looking at laboratories and insurers in terms of stakeholders, as we call them, in getting genomic medicine implemented. The fourth one was on physician education, on federal strategies, one on global leaders that some of your colleagues came to, genomic clinical decision support, a review of all of our programs, one on bedside back to bench, another on pharmacogenomics and clinical implementation, and then we have one coming up in just 2 months on genomics and risk prediction. I would mention this one in particular because it's where I met your colleague Sarah Kamethan and Professor Watson. And we heard at that meeting this very exciting work that was going on then, and I can tell you, I didn't just put these slides in for this talk. I include this slide in almost every talk I give on genomic medicine because we were so inspired by the work that's going on here. This pharmacogenetic card that you developed, I actually carry one with me. It's not mine, it's a blank one, but just as an example to places around the world that are trying to implement genomics, we try to make the point this doesn't have to be complicated. It doesn't have to involve very sophisticated machinery or computers or anything. Something as simple as a card like this can save somebody's life. And in this example, identifying a patient who's at risk for carbamazepine-induced Stevens-Johnson syndrome. I should note that once we had that initial meeting where we heard about this, we had a subsequent follow-up meeting specifically on Stevens-Johnson syndrome toxic molecular isolate and have identified some research efforts in that to move forward. And we've been very delighted to see the slide that I got from Sarah Kamethan describing how you noted that you had a terrific epidemic going on. You had some brainstorming about what to do about it, implemented some testing, demonstrated cost effectiveness, and then implemented nationwide testing. I'm hoping while I'm here to get updated data from December of 2018 so that you can complete this slide and I can show it with pride wherever I go. These genomic medicine meetings are posted on our webpage for the Division of Genomic Medicine. If you click on this, it will give you the list of them all the way back to the first one. This is the most recent one that's been completed. If you click on that, it will show you the slides that are accessible. You can get a video. We actually video record all of these meetings, and you can access those, as well as the meeting summary and executive summary. So I encourage you, if you have any interest, to take a look at these. They're usually quite topical, and they help us to identify important research directions. And we often publish papers from them. Here's one from the very first one that we held looking at national efforts in the U.S. in implementing genomics in clinical care. Another one trying to figure out how best to characterize genetic variants for use in care. This was the education meeting focusing on professional societies' roles, global implementation, as I mentioned, that's the 6th meeting that we held. This one published in Cell on linking together laboratory and clinical scientists, and then the clinical implementation of pharmacogenomics. So quite an active effort. I think at this point I'd like to turn to some of the research programs that have come out of these planning meetings. So one of the things that we do at these meetings is to identify research questions, and then it's our job at the NHGRI to go back and determine how can we design studies to actually address these things. So what you see here are sort of a list of the major programs that we have. These are all acronyms so that they fit on the slide. Undiagnosed Diseases Network diagnoses rare and new diseases by expanding what had been an existing undiagnosed diseases program on the campus of the NIH. This now involves about 15 centers across the country trying to develop appropriate ways for these patients to be diagnosed rapidly and get into treatment. You'll notice that there's a significant amount of money, $237 million, committed to this over the 10-year period that it's been running. So it's a really significant effort, the largest one that we have. The EnSight program, Newborn Sequencing in Genomics and Health, was an effort to explore uses of sequence information in the newborn period. I'll talk a little bit about that. A much smaller program over a 5-year period. The CSER program, Clinical Sequencing Evidence Generating Research, is to generate evidence of the clinical utility of sequencing in a number of different sorts of clinical settings, undiagnosed diseases, intellectual disability, pediatric cancer, things like that. And again, a pretty large program over a 9-year period. The Electronic Medical Records and Genomics program uses biorepositories with electronic medical records for genomics and assesses the penetrance of clinically relevant genes. The Ignite Implementing Genomics in Practice program develops and disseminates methods for incorporating patients' genomic findings into their clinical care. The Clinical Genome Resource, or ClinGen, is to develop and disseminate consensus information on genes and variants that are relevant to clinical care. We also have a number of what we call investigator-initiated programs. These tend to be smaller, and they come from individual labs or universities that have a good idea and propose to do a research program that then is peer-reviewed, and those that are viewed as scientifically meritorious we then fund. And they are involving a variety of areas, adverse drug reactions, clinical sequencing, a number of different things. And we also have a small training portfolio to try to raise up the next generation of genomics investigators. We see this genomic research, at least our portfolio, on a spectrum. So if you think of the depth of patient characterization, going sort of down the y-axis there, and the breadth of implementation across the x-axis, plotted arrow, we see the Undiagnosed Diseases Network as really having a very sharp patient focus. Very often these patients are admitted to the hospital for a week and really very intensively investigated. Similarly, the newborn sequencing program also has a very direct focus on individual patients and different models, different approaches to doing this kind of work, and sort of try to figure out which one works best. The CSER program, the clinical sequencing program, is a little bit further along in terms of breadth of implementation, not as much depth of patient characterization, but it looks also at the impact on clinicians and laboratories and families and others of genomic information. And then our Emerge and Ignite programs are much more along the path of systematic implementation, evidence generation, and really how this information is used and works in a hospital system. And then sort of overarching them all is the ClinGen program, the Clinical Genome Resource, because it's designed to interpret variants and be sure that everyone is using them in the same way in clinical care. And I'll give you some examples of each of these, starting with the UDN. So the Undiagnosed Diseases Network has a number of sites, you can see them here, around the country, including the NIH. This is a paper published by that group about a year ago, looking at the effect of genetic diagnosis on patients with previously undiagnosed diseases. So this is the experience from the first 1,500 applicants to the program. Of those, about 380 were accepted and underwent a complete diagnosis. So unfortunately, there are many more patients who want this resource than we are able to admit into it. And there are fairly strict criteria for selecting patients. And you might want to, as you develop the programs in this area, I'm sure Professor Borsch uses similar criteria to determine who to take into the program and who may not be likely to benefit from it. 35% of the patients who had undergone a complete evaluation received a diagnosis, with 31 new syndromes identified. You can see here the diagnosis by any method occurred in 35% or 132 patients. Interestingly, of those 132, about 30 of them did not need genomic information to make a diagnosis. Just having experienced clinicians carefully reviewing the records or doing a little bit of extra genetic testing came up with a diagnosis in those patients. But there was a fair number of them, about 98 or 74% of those in whom a diagnosis was made, where sequencing information really contributed to the diagnosis. And sometimes that was even in patients who had sequencing done previously, but perhaps it was reinterpreted or there was additional information provided that could be interpreted. The newborn sequencing program I described to you, it involves 4 different groups, as you can see here. I won't talk about all of them, but I'll pick up 2 at least. So this is a randomized clinical trial from Stephen Kinsmore's group in San Diego, where they were studying infants in neonatal intensive care unit, particularly infants who were not there because they had infections or because they were premature, but something else was wrong with them and no diagnosis could be made. It was a fairly small study, 65 infants, less than 4 months of age. They were studied in trios, which means that the infant and both parents were sequenced. They made 31% of these children who received whole genome sequencing had diagnoses made, compared to just 3% of those who did not have the whole genome sequencing. And the time to diagnosis was really quite dramatically different. So within 2 weeks in those who received sequencing, and much, much longer for those who did not. And you can see these graphs here that show how quickly time to diagnosis in days in the intensive care unit, and really all of these children had their diagnosis within 90 days, whereas it took quite a bit longer for the smaller number that received them with standard testing. And the age of the child was quite a bit younger in those who received diagnosis. And this is particularly important in this age group because many of the conditions that these children suffer from lead to irreparable damage in this very critical period of development. And if one can identify the illness, and perhaps if it's a nutritional deficiency or metabolic abnormality, you can implement interventions to prevent that. That saves really a lifetime of disability and heartache. So very exciting findings from that program. A similar program as part of that from the Harvard group looked at 159 newborns. Some of them were well, and some of them also were in their neonatal intensive care unit. 9% of them had risk of childhood onset diseases, 5% had important pharmacogenomic abnormalities, and close to 90% had recessive childhood onset disorder carrier status. So they didn't have the disease, but they were carriers for diseases. And you can see actually that 47 out of the 159 had even 2 variants that were associated with recessive carrier status. There's a lot of debate as to what to do with this information in an infant, because it's probably most important when they become of reproductive age. But it is important and useful for the parents to be aware of if they're planning future children that they may want to be concerned about. Our electronic medical records and genomics, or eMERGE network, is one of the more complex of the programs that we have. It involves genome-wide association testing, electronic phenotyping, methods for obtaining consent, community consultation, data privacy efforts. We've recently added pediatrics and pharmacogenomics to it. There's a major clinician and patient education component and clinical decision support as well. One of the key things that the eMERGE network does is to develop methods for using electronic medical records for genomic research, hence the name. And as you can see here, one of the things that they do is to try to determine true cases of disease based solely on electronic medical record information. So this is more than what we call billing codes in the U.S. The way that hospitals are paid and doctors are paid is they make a diagnosis, that diagnosis gets assigned a code, and there's a certain amount of money that goes along with that. Those are called billing codes. As you might imagine, they're sometimes not the most accurate things in the world because being capitalists that we are in the U.S., sometimes the diagnosis might be sort of up-coded a little bit to get a little bit more reimbursement from it. But those codes, as well as something called CPT, which I've forgotten what it stands for, but it's basically a procedural code. So those can be incorporated into the diagnoses or the phenotypes that eMERGE comes up with. Clinical notes using natural language processing, lab and test results, again with natural language processing, and medications, electronic prescribing, etc. And you come up with your true cases. And these are then, these algorithms for these various conditions are then stored in the phenotype knowledge base. Again, an open resource. One of the jobs of NHGRI, which actually came from the Human Genome Project, is to produce resources for use in genomic research. So these are all available through the PKB knowledge base. And you can see a list of just some of the A diagnoses here. Let's start with A. And then sort of where they were developed and what's involved in them. And if you click on one of those, it will tell you a little bit more about, say, atrial fibrillation, how it was diagnosed and developed, and what pieces of information are used to define people who have and don't have the condition. And then you can even look at how they were implemented. So this is cataracts at the Marshfield Clinic in Wisconsin, and they identified patients based on the EMR algorithm compared to expert clinician review and classified a couple of thousand patients into true positives, true negatives, and then those that were sort of mistakes. So people that the algorithm said had the disease who clinicians felt did not, and vice versa. But you can see that what we call the positive predictive value was quite high, 98% of the time. If the algorithm said that a patient had the patient had the disease according to the clinician. And then similarly, for people who did not have disease, pretty good positive predictive value. So we're quite satisfied with these algorithms that get published. They do get transported to other eMERGE centers to be sure that they can. It's not just something that's unique to the Marshfield Clinic or the Mayo Clinic or whatever. And these are available for reviews. This is a recent paper from the eMERGE network that was looking specifically at how do you take the massive amount of genomic information that you get, 3 billion base pairs, a lot of information. How do you integrate that into the medical record and use it in clinical care? And one of the challenging things that we did in the eMERGE network was to actually have two different genomic laboratories do the genotyping, because we wanted to sort of challenge them to come up with standards between the two of them and kind of make each other better, which happened, which was great. So the Baylor College of Medicine was one lab, and then Partners Healthcare in Boston was the other. They not only need to provide data for research, which goes into these things of de-identified data without patient identifiers for research, BAM and BCF files, BAM being very large sequence data files that are very difficult to store and manipulate. The BCF's variant calling files being the smaller files that are typically used for association research. But then they also needed to provide this information back to the clinicians at these various hospitals, so Brigham and Women's Hospital, Cincinnati, and Geisinger Clinic, and others. So this group provided their data back to these 4 hospitals, and the Baylor group provided it back to these 6 hospitals. But because this is one program, those clinical reports needed to be in a common format. And getting these 2 groups to agree on a common format was a real challenge. But we think we ended up with a product that was better than either of them sort of by themselves. And this structured data format is available again for open use. So please try to reuse some of these things. You don't need to reinvent stuff that's already been done. And we feel this is a valuable resource. We hope you will too. We also, in the eMERGE program and several of our other programs, try to identify, at least keep up with professional society guidelines for identifying secondary, what we call secondary findings or abnormalities that are detected in the genome sequence that probably have some impact on people's health. Trying to figure out which of those to report has been a challenge for the past 10 to 15 years. And relatively recently, the American College of Medical Genetics and Genomics really sort of jumped into this and came up with a consensus approach that one can argue with, but it seems to be sort of standing the test of time. To identify 50 variants in 59 genes that are typically associated with cancer syndromes or cardiovascular syndromes, and be sure to report those back to patients. So there's a list of these. If you haven't seen this paper, I highly recommend it. For example, the hereditary breast and ovarian cancer, BRCA1 and BRCA2, that gives you a fair amount of information about them, including where the OMIM, the Online Mendelian Inheritance in Man resource is that describes them in much more detail, the inheritance, et cetera, and what kind of variants to report. This is a list of all of those 59 genes. It's in that paper, but I just extracted it for you here. And you can see it's focused primarily on things that have significant burdens of illness, so morbidity and mortality from cancer syndromes, as you can see up here, and then cardiovascular syndromes, most of these leading to either sudden death or heart failure, and then a few metabolic syndromes as well. The IGNITE, or Implementing Genomics in Practice network, is shown here. These were clusters of centers. You can see there's a group here in Tennessee, another group in Indiana, a group in Florida. What we asked these centers to do, all of these were expert centers that were leading these groups, and we said reach out to groups who have not done genomic medicine at all, and try to find out what it takes to get hospitals that may be, very resource limited in inner cities. So there's a number of them in the Bronx in New York. There are several of them in Wisconsin and scattered across the country. Find out what it takes to get them to be able to implement genomics in a way that's efficient and effective for them. Very much like a pharmacogenomics card, if that's the kind of way one can go about it, or if you can do it in a more fancy and sophisticated way, that's great too. So the IGNITE network was designed to expand and link existing genomic medicine efforts, develop new collaborative projects and methods in diverse settings and populations, contribute to the evidence base regarding the outcomes of incorporating genomic information in clinical care, and defining and sharing the processes of genomic medicine implementation, diffusion, and sustainability. One of the seminal findings from that program was one of the first demonstrations that actually using a pharmacogenetic variant in the care of patients who are undergoing coronary catheterization and receiving stents. There was strong suggestion that knowing whether these patients were able to metabolize the antiplatelet drug clopidogrel that they were given after they received their stents would have an impact on their care and their outcomes. But there really wasn't much in the way of evidence for that in the United States, and we're very sort of evidence-driven, sometimes a little too much so, I think. But at any rate, what the IGNITE group did was to get together and review about 1,800 cases, genotype those people, and determine which drugs they received and what their outcomes were. So what you can see here in the red line are the patients who had a loss of function variant in this gene, CYP2C19, that prevents them from being able to metabolize the drug clopidogrel, so they get no benefit from it, essentially. And those who had that variant and received clopidogrel had a much higher rate of events, major adverse cardiovascular events, much higher than those who had the variant and received an alternative drug that does not require them to be able to activate it, to metabolize it. So this seemed to us to be pretty strong evidence. And in fact, when we compared those people who had the variant in their gene to those who didn't have the variant in their gene, they actually did a little bit better if they received the alternative drug, suggesting that maybe there are some other variants that need to be identified. The Ignite investigators, because we are so focused on being sure we provide these methods to the broader community, has developed something called the Spark Toolbox. So there's a section in it for clinicians, sort of how to get started implementing genomic medicine. You can click on CYP2C19 clopidogrel, and if you do, it will open up a whole host of options for you to learn about how these implementations were done across the 6 network sites in Ignite. If you look at, for example, genotyping resources, you can open that and there's a list of them, and each of them is described in fairly large detail. This particular one from the University of Pennsylvania using the Spartan system. There's a whole lab manual here, and it's again free for the taking if you're interested in pursuing. You can also look at implementation workflows. So how do you actually get this into, when the patient is seen, when the blood is drawn, when they receive their catheterization, when they have follow-up, etc. And they have a very nice flow chart where the patient comes in, undergoes their coronary intervention, and various things that happen then. So you don't have to start from nothing. You can actually use this experience and modify it. Maybe your lab doesn't come in at this stage. Maybe it comes in earlier, or maybe you don't have a pharmacist, etc. But it's a place for you to start. And then there are other useful resources as well. So again, these are all things that we try to make available to kind of make best practices known to the community, and by looking at these and using them. Clinical Genome Resource, or ClinGen, is as I said, sort of the overarching resource that we're building basically to interpret genetic variants. So you can sort of see here the number of genes that they've interpreted, the number of expert groups that are doing this work, the number of variants reviewed, etc. There's a wealth of information on this website. They try to answer basically 3 questions. Is this gene associated with disease? That's sort of a clinical validity question. Is the variant in this gene causative of disease? That's a pathogenicity question. And then is the information knowing that you have this variant in this gene, is that actionable? Can you do something about it? So that's clinical utility. So there were many groups when we first started this effort, many groups doing this kind of work all on their own. So they were all sort of separated. They would have meetings where they'd sit down and review whatever evidence they could get from the literature. They would all make decisions as to whether a gene was causative and a variant was causative, and they would basically come to the same conclusions, but all in isolation. And we said, that's silly. We need to have a coordinated effort for doing this work. And because the genome is so big and there are so many genes, can we divide it up somehow so that you don't have to review a variant or a gene 20 times. You can review it once according to a standard framework. So one of the biggest jobs they had to do was to create the standard approaches to curating these genes and variants, and then develop an authoritative resource to define the clinical relevance, establishing a semi-quantitative framework to assess the strength of evidence, promote public sharing of variant interpretations, improve and disseminate rules for variant interpretation, resolve conflicts between laboratories, and engage experts in a systematic consensus-driven interpretation. There are currently, actually there's probably more than this now because this is a 6-month-old slide, but in October there were 850 researchers and clinicians from 27 countries, and you can see them all here. There's not anyone there yet, but hopefully there will be soon. And one of the very exciting things that they did recently was to basically review 21 genes that had been reported as associated with sudden cardiac death or Gala syndrome. We call this de-implementation for the reason I'll tell you in a moment. What you can see here is a list of all these genes. You certainly don't have to be able to read them. And then these are the panels in various countries that include these. When you're testing someone for risk of sudden cardiac death, you can see that Australia is this reddish color here, and it includes these genes plus a number of others. This one is Canada, I think, and it also includes several. And there are some genes that are on just a few panels, and then there are others that are on almost every panel, including SCN5A, which was the original gene identified as a risk factor for Gala syndrome, which is the most widely recognized syndrome leading to sudden cardiac death. What ClinGen did was to systematically review each one of those genes and look at the evidence for each of them. They had 3 different curators, expert curators, looking at them all. And you can see that all of these genes, even for 3 curators, none of them got up to the highest level, and none even got to the moderate level. So all of these genes had really limited evidence for pathogenicity, and only one had very strong evidence for pathogenicity that all 3 curators agreed on, and that was that original gene, SCN5A. All the rest of these are disputed or refuted, and they are now no longer recommended to be tested. So instead of 21 genes, we now have just SCN5A in sudden arrhythmic death. So this information has gotten out to the people who make these panels, and they're changing their panels, because one thing you don't want to do is decide that somebody is at risk for sudden death when they may not be. That is then a shadow that hangs over them, and they very often will end up getting an implantable defibrillator, which is not a small task, a small thing to do to someone. These results go into a database called ClinVar. It's the database on which ClinGen is built. So we refer to the information here for classifying variants. And this is a description of it. This is a screenshot of what it looks like. If you open up, sort of search on a gene and a variant, you get information like this as to who has seen it before and what they thought. Did they think it was pathogenic? Did they think it was benign? And what was the evidence that they used to determine that? And you can see here there were 2 groups that agreed, so that's consistent across multiple sources, and ClinGen actually gives a rating to that, so that's a sort of a 2-star rating, which is useful to know when you're trying to determine which variants might be associated with disease. So you can see here the likely pathogenic rating there. So this is a huge effort. ClinVar actually has nearly 500,000 unique variants submitted from 1,100 submitters across 67 countries. So a huge effort going on, and even here, if we kind of blow up this area here, you can see we're delighted that Thailand is there. Thank you very much for depositing data in ClinVar. It's extremely important for us all to be able to interpret our variants. And if you take a look at who submitted what, this is the submitter's page from ClinVar. It just lists the group and actually has a link to their website, the way that the review was done, the total number of submissions, the last update of those. And so if we look at the Southeast Asian area, you can see in Singapore, there's a group that did a very nice job, 1,100 genes submitted, although they were a little bit older in July of 2012. Your colleagues in Malaya, only 3 genes, so not quite so many, but at least they're trying. But then we look at Thailand, and we really only have 1 gene submitted. And so I kind of ask, why is that? And would ask you, and maybe stimulate you a bit, to please submit your data to ClinVar. It is so important, because there's no way that we can possibly interpret all of the variants that occur in 1 or 2 or 3 people in one country. But if we can address this in a global way, we will be able to interpret these. And so it's crucial to really deposit these data. So I would just close by noting genomics is global. It takes all of us to understand each of us, and to the degree that you can contribute to that, you're participating in the global research community, and the benefits will be yours as well as everyone else's. So with that, I'd like to stop and thank my colleagues at NHGRI and our investigators, and I'll be happy to answer any questions. Thank you very much.
Thank you very much, Dr. Teri Manolio. I think it's time for question and answer. So we have a 15 minutes for question and answer.
Good afternoon, Teri. My name is Manol from Ceylon Hospital. Thank you for your comprehensive information on genomic medicine. I have 2 questions. Probably there's I don't know, some kind of correct answer. I don't know what it's going to be. The first one is when we hear some people in Thailand say we are in the middle of, we don't know if we are wealthy enough to invest in implementing high technology into the healthcare, for instance, or we still need to focus on something that very basic and useful to everyone. At one point, some people say this genome technology is kind of very expensive and fancy. So we should focus on something that's very basic first. And on the other hand, we want to move forward to implementing this technology to benefits in the health system. I don't know, should we pursue on something that's have the great impact first, for example, pharmacogenomics, and stick on that, or should we try to expand into other area that in the short period would not see any immediate benefit, but it would be very good in terms of investment? That's the first question.
So could I just stop you there?
Okay.
Because my memory is short. So that I think is a constant tension with any new technology. So you constantly say to yourself, should we introduce MRI, magnetic resonance imaging? Should we, be using this new expensive drug? And that's a balancing that has to be done with a vision to the entire country's health needs that I'm not really qualified to comment on. What I can say is that there are clearly instances where genomics does help and is cost effective. I think neonatal sequencing is one of them, as is pharmacogenomics for certain indications. And so obviously starting on the high value things is a good place to start. The challenge is that you really can't do those in an efficient way unless you develop the infrastructure, and that infrastructure is much broader. It isn't really an either or, it's both. I'm sorry, I can't give you a better answer than that.
And the second question is, since you have presented us a lot of the program that's very useful in terms of implementation, but sometimes we come up with something that the data shows some conflicts with each other. For example, that you mentioned about the data on the implementation of 21 gene panels for certain death. On the other hand, when you see the ACMG recommendation on secondary finding, you still see a list of some genes that have the overlap that we need to report. So when there are two new information that emerge, should we, as a clinical lab, how do we proceed on this?
Yeah, and that's, again, another struggle. So you're asking very difficult questions that I don't have good answers to. But I think, one way to proceed is to look toward consensus building. So many other people are having the same experience that you are. And we like to say in the States, 2 beds are better than 1, and many beds are better than 1. The way the ACMG approached this was to try to bring together experts in the field and ask their opinion, because that's all we have right now, and it's a place to start. And so what's going on in programs like eMERGE and others is really, okay, if we implement these, what happens? Because that experience hasn't been gathered. And so I think this is a place to start where it comes to research, and not an end in itself.
Dr.Teri Manolio, thank you so much. That was very informative, very relevant to us. We are going to launch Genomics Thailand, and this is very informative. Thank you so much. I'm Barasak from Chulalongkorn University. I have this chance to lead one of the 5 areas of Genomics Thailand, which is rare and undiagnosed disease, which is similar to the UDN. My question is, you have a budget for up to diagnosis, and some of these is going to be actionable, but I guess you don't have budget for the real action. For example, if you diagnose a patient with a rare disease, but I guess you don't have budget to treat them, and it's even problematic if that disease has a drug, but it's so expensive. Someone said, well, just don't do it. if you do it, it's like open a can of worms. What is your suggestions in probably our context? In Thailand, it's even more. Right? You don't diagnose this child. Everyone is fine, accept it. Okay, maybe it's because of his past life or whatever. But when you diagnose this child with a certain disease, and the parents and family know that, come on, there is a treatment. The government should provide that treatment because we have a universal health care scheme, which our politicians advertise that everyone can be cured of any disease without paying anything. So what's your suggestion?
Yeah. So in the U.S. we don't make any promises like that. And we unfortunately have a healthcare system or a lack of a healthcare system that does not provide the kind of universal care that yours does. It's a much more difficult equation, I think, for you. We recognize that patients who are not diagnosed continue to use medical resources, so they're continuing to search for a diagnosis, and that costs money. When you have a patient in the neonatal intensive care unit, I don't know what it costs in your country. In ours, it's about $15,000 to $18,000 per day that they're there, where the sequencing costs about $3,000 if you're doing the parents and the child. So that cost benefit, is pretty clear. The question of treatment is another one that comes up in epilepsies where there are very expensive drugs, and cancers for sure, in retinoblastoma. There are a number of different settings like that, and I'm not smart enough to be able to make those decisions. But I think if you have people continuing to use medical resources to make a diagnosis, you're a step ahead if you at least make the diagnosis. Then you have to make a decision about how to treat them and whether to treat them. I think, one of the sad things right now is that many of these conditions we make a diagnosis, but there is no treatment. We hope to find those treatments, and we hope the cost of them will come down. But even in the intensive care unit children, about half of the diagnoses that the King's More Group makes, there's nothing we can do about this. This child is going to die. What that does for the family is allow them to sort of, I don't know if you've ever been in an intensive care unit. It's a horrible place. I mean, there are buzzers and bells going off. There are people everywhere. It is not, a place to try to get better. And for families to at least be able to spend the time they have left with their child in a quiet setting, perhaps outside the hospital. We have hospice. I don't know if you have that here. Take them home even. And that's, I think, a gift for the family that we can provide. And so on balance, what's the benefit? I think that each of us has to answer that for ourselves, and the policymakers have to answer it as well. That's what would be my answer.
Very helpful. Thank you so much.
Hi, I'm Terry. There are 3 questions to ask, maybe one at a time.
One at a time.
And the first one probably might be like a complaint. We have like a global leader in Genomic Medicine 2014, if you remember, that me and Suratame participated. And then during the meeting, we come up like we should focus on pharmacogenomics. And it turned out it doesn't fade away from the NHGRI radar. I'm not quite sure. And now it popped up to be like a global leader or global pharmacogenomic lead by UK group, Manolio, for example. Can you comment on that?
So when you say it faded away, do you mean Stevens-Johnson toxic vector molecular?
Right.
So one of the challenges that we have, I mean, it's a good problem to have, I guess, is that condition is very rare in the United States. And so we had a whole symposium, as you know, you were there and spoke very eloquently at it. We could not convince the leaders of the various research groups that would be responsible for this, that this was a problem worth investing in, because in our population it's about 1 in a million and it occurs. So we weren't able to go that route by what we would call an institute-initiated program like Ignite or Emerge, where we say, we're going to go in this direction, please come along with us. what we did do was to say we think more research is needed in this area in the U.S. and we invited applications and we got some, particularly from the Phillips Group at Vanderbilt University. So we're funding that. It's a much smaller effort. But we did recognize that there are other problems in pharmacogenomics that are very important in the U.S. And we thought we would sort of go at those first. And if we can convince the policymakers and the decision makers that there are 2 or 3 or 4 or 5 gene-drug combinations that are worth testing for, it's much cheaper to actually test them all than it is to just test 3 or 4 or 5. So we're hoping to get in sort of the back door by testing the ones that are most important in the U.S.
So in U.S., when you talk about pharmacogenomics, perhaps you mean the cytochrome or the dose adjustment instead of HIV.
Yes. Yes, yeah, exactly. So it's my dream. So we're about to embark on a clinical trial that basically tests for opioid abuse, depression, and pain management. And that will be a panel of genes, very much like the European trial that's going on currently. And if we show that that is effective, then I think we can make a very strong argument for testing a much larger panel of gene-drug pairs.
And way back, if you remember, that the Alan Schussler and of course recommend mentioned that they tend to eradicate Stevens-Johnson syndrome in Thailand, and actually we are doing it.
Yes. Well, and that's why I show your slides. I mean, I'm thrilled, and I try to make this point almost anywhere I go. This is a problem that is very strong in Thailand and in Southeast Asia, and so you can show an effect much easier than we can. So we're going to build off of your results for that. You can build off our results for clopidogrel.
Second question. For the when you talk about sequencing in US, you mean whole genome or exome?
Primarily, when I mention sequencing, it's whole genome.
Whole genome.
Mainly because, as we've seen from genome-wide association studies, 90% of the associations are not in the exome, so they're outside of the protein coding regions of the genes. We don't understand those right now, but we can use that information in polygenic risk scores and other things. So yeah, focusing more on the whole genome.
And for the rapid whole genome sequencing for newborn, you're talking about the trio, and who paid for that? The reimbursed?
It's beginning to be reimbursed because of data like the Kingsmore data. So there are a couple of insurers who are large insurers who are now starting to pay for it because they recognize it's a lot cheaper to do the trio sequence than it is to pay for a day in the intensive care unit. So it's beginning, but it's slow. Our progress is just so slow. Sorry.
The third one, the short one, is a clean one. The main reason that we never submit because we are lazy. He is lazy. And I promise that from now on, because we have it maybe on the weekly basis that something will be the phenotype and variation.
No, that would be terrific. And it's not a trivial process to submit to ClinVar. I know that. I mean, it's a pain in the neck and it takes time and it's not reimbursed. We try to make the process easier. There's sort of a submission wizard that's been built. It's a bioinformatic tool to do that. But you are going to see variants that nobody else is going to see for years or decades. And if we can interpret those based on your data, that would be terrific. Yes. Oh, absolutely. Exactly. Yes. Yeah. And you get cited as being part of that. There was mention earlier today of joining the Global Alliance for Genomics and Health, the GA40H. That's another way that you can have influence on the entire field. You can help set the standards and participate in that research.
I'm Duntanom from Department of Medical Sciences. If we talk about pharmacogenomics, as far as genes of each individual can be already known, so it means that if those genes are responsible for each disease, it means that there should be more than one drug to be selected or many drugs to be selected to best suit for treatment of each disease, right? But as we know that nowadays most of the drugs are ready-made drugs, so I have an idea about how far of the world of drug development nowadays, how far of those people to make so many new drugs to serve for the worldwide project of this genomic medicine.
So I'm sorry, your question is, are people developing drugs to treat the individual indications that we find? Well, to some degree, unfortunately, in the U.S., when they make a drug, particularly for a very narrow indication, they tend, this is drug companies in general, tend to say it's very, very expensive, we're not going to be able to recoup the costs of development, so they charge outrageous amounts of money for them. Up to $1 million a year, which in my view is just criminal, but they didn't ask me. So something that we need to be able to do is to develop drugs on a much shorter timeline and at much lower cost, and then provide them to our patients at a reasonable cost, not at something that will either bankrupt them or bankrupt the healthcare system. We don't have a solution for this yet. It's a huge problem. I'd love to hear how you're tackling it here.
Any questions? Okay, I think it's time for giving a token of appreciation. Thank you very much, Dr. Teri.